Hi, everyone. My name is Jifang Zhao. I'm a senior data scientist at Olaris. Olaris is a metabolomics company that specializes in developing diagnostic tools. By merging metabolomics with machine learning, we are enhancing the capability of disease diagnosis, sequencing therapy, and monitoring adverse effects such as ischemia. Today, I want to share some exciting research we have been conducting on the metabolite donations that take place during donation of circulatory death, or DCD. Heart perfusion. As many of us are aware, there is a significant shortage of organs availability for transplantation across all types of solid organ transplants. This scarcity is particularly prominent in heart transplants, with approximately 4,000 individuals left awaiting for a transplant last year. One potential way to address organ shortage lies in DCD transplantation, which has the potential to increase the number of available organs by around 30%. However, despite its potential, DCD is currently underutilized, with adoption rates barely exceeding 3%. Several factors contribute to the hesitance toward DCD. DCD is expensive, it is FDA approved just years ago, and more importantly, there is a lack of understanding of its process comparing to traditional transplant method, DBD. In typical heart donation, a brain-dead patient's heart is harvested and preserved on ice for quick delivery with a 4-hour window. On the other hand, DCD heart is harvested after the heart stops beating naturally within 30 minutes and revived in a perfusion machine, which extends safe transport to 6 hours. Concerns surrounding DCD arise from the process' unique stages. Wall ischemia, where the heart lacks sufficient oxygen and potential ischemia perfusion injury from reconnecting the heart to a perfusion machine could cause damage. This uncertainty makes physicians hesitate to transplant these hearts. The lack of reliable biomarkers in DCD poses a significant challenge. Studies that demonstrate the only biomarker available, lactate, is inconsistently correlated with long-term outcomes and the quality of the heart donated. This highlights the need for better understanding of what actually happens during DCD. In this research, our primary goal is to gain a comprehensive understanding of DCD. Specifically, we are going to identify changes in metabolites during DCD perfusion and then link altered metabolites to biological pathways of interest. This understanding will serve as the initial step toward development of new tools that can provide greater confidence and empower decision-making for heart transplantation procedures. In our research collaboration with Massachusetts General Hospital, we had access to 63 human plasma samples from 20 DCD hearts, involving 19 hearts. Notably, one heart was transplanted into a recipient who unfortunately passed away after 2 weeks due to a stroke. The same heart was then used for another transplant. For each transplant, we collected 3 samples, a baseline sample taken immediately after placing the heart on the perfusion system, a median time point sample collected during the transplant process within 2 to 4 hours on the perfusion system, and a final sample obtained right before the heart transplant. The endpoint sample timing varied based on transportation distance, typically occurring 2 to 6 hours after heart harvest. At Olaris, we use our Cerebral platform to analyze human biofluid samples by combining NMR spectroscopy and machine learning for metabolite detection. Traditional 1D NMR provides a comprehensive and quantitative analysis of metabolites, with each peak in the spectrum representing specific metabolite and its concentration. However, Chemical shift overlap limits the number of metabolites that can be accurately and unambiguously quantified using 1D NMR, which was more significant in the perfusate samples due to intense signals from compounds in the priming and Menon solution. In this study, we employed non-uniform sampled multi-dimensional NMR technologies to enhance the accuracy of quantification and identification of metabolites. This approach spread the 1D NMR across multiple dimensions, enabling more precise analysis. Using technique, we successfully identified numerous metabolic changes that occur during the DCD process. At first, we conducted two analyses. In the first analysis, we studied the metabolic alternation when the DCD heart get connected to the perfusion machine. We examined metabolite levels at various time points and compared them to the baseline. After rigorous statistical analysis, we identified many significantly changed metabolites. These changes, as visualized in volcano plots, clearly illustrate differential features discovered. We further gain insights from these changed metabolites using unsupervised clustering, as seen in these heat maps. Remarkably, the baseline metabolites showing pink differed significantly from those during machine perfusion or pre-transplant stages. This clear separation suggests a significant metabolic change once the heart are connected to the perfusion machine. As an interesting pattern emerged when comparing baseline samples to various time points, we constantly noticed changes in 9 NMR resonances at each stage. Remarkably, these resonances also changed from baseline to pre-transplant, emphasizing this metabolite's importance in the DCD process. In the second analysis, we explored metabolic changes over time at various time points, comparing different sample sets. Interestingly, we observed significant variance in many metabolites over time. Each time point data differed from later ones, implying ongoing changes during perfusion. To understand these metabolites better, we focused on 13 consistently altered resonances from an initial set of 30. Multi-dimensional NMR, particularly HSQC spectra, help us identify changing metabolites using NMR resonances. By matching the chemical shift of identified resonances with reference libraries and verifying with spiking experiments, we ensured that identified resonances truly correspond to changes in metabolites. Among the identified 13 resonances, we found glycine, alanine, arginine, 2 peaks from gluconic acid, and 2 NMR resonances from unknown metabolites. Additionally, we observed 6 resonances that match to potential fatty acids. However, due to chemical shift overlapping, it is challenging to identify these fatty acids using NMR. Therefore, we currently refer them as potential short or medium chain fatty acids. When we plot the choreography of these top metabolites, the dynamics of these metabolites revealed an interesting trend. The potential fatty acids constantly decreased during perfusion, while the level of amino acids and sugars increased. This suggests shifts in energy metabolism and nutrient utilization. To ensure the reliability and validity of our findings, we conducted a series of studies to address potential factors that could influence the observed changes in metabolites during the DCD process. In DCD process, a liter of blood from donor is mixed with a liter of priming solution and circulated through the heart while it is connected to the perfusion machine. A mixture of nutrients and amino acids known as Mannan solution is constantly dripped into the blood mixture during perfusate to maintain the level of lactate. It is possible that the observed changes in metabolites could be attributed to hemolysis or compounds in the Mannan solution. Hemolysis occurs when red blood cells break down, releasing their contents into the surrounding fluid, which can impact the composition of the samples. During our investigation, we observed a visible sign of hemolysis in the later time samples. These samples appeared darker and redder in comparison to baseline samples. We conducted an experiment to isolate the effect of hemolysis, in which we circulated blood through the system without the presence of a heart for 3 hours to assure the influence of hemolysis on our findings. We then closely monitored and compared the level of the 13 resonances during this period. Remarkably, the results revealed that fatty acids resonances, along with alanine and glycine, remain unchanged over the course of the experiment. This finding strongly suggests that hemolysis did not significantly impact these metabolites. In addition to considering the influence of hemolysis, we also examined the impact of the Mannan solution on the observed metabolite changes. During perfusion, a Mannan solution is constantly added to the circulating blood to keep the heart healthy. This solution contains known compounds, including amino acids such as alanine and glycine. To untangle the effects of Mannan solution from underlying biology, we conducted an experiment using an orthogonal pig model. In this model, we placed a pig heart on a perfusion machine, but instead of using the regular Mannan solution, we utilized a chemically defined solution known as STEAM, which does not contain amino acids. The result of this experiment was truly exciting. We found that 10 out of 13 resonances that changed in human samples also show changes in pig sample. Furthermore, the trends observed were nearly identical. The fatty acids displayed a decrease in their levels, while alanine showed a substantial increase, similar to what was observed in human samples. Through these comprehensive analyses, we were able to confirm that the top 13 resonances we identified were not influenced by the Mannan solution and hemolysis, but rather reflected underlying biology processes. To understand the biology behind the observed metabolite changes, we conducted a network analysis, which involves examining the correlation and structural similarities among the top metabolites, as well as identifying their connections to different biochemical pathways using a nearest neighbors map. This analysis revealed relationship among the metabolites. For instance, arginine, alanine, and glycine, which showed alternations, were found to be precursors or substrates for numerous other metabolites. This suggests that their increased level may impact the flux through these pathways, potentially influencing various biochemical processes. Investigating these pathways further will be a critical focus of our further research. In every analysis, a clear rise in alanine levels has apparent in both human and pig hearts. This finding is noteworthy since currently we only have lactate as a reliable biomarker for events during transplantation. However, our studies show that while lactate level remains stable, alanine experienced a significant increase. In the pig model, the surgeons intentionally allowed lactate level to rise, while in real transplants, we actively remain low lactate level. Despite this manipulation, the increase in alanine suggests the underlying biology of ischemia and ischemia reperfusion injury could still be occurring. This highlights the potential of adenine as a more sensitive indicator of ischemia and ischemia reperfusion injury. Furthermore, existing literatures already provide substantial support for the role of adenine in these processes. These findings not only contribute to our understanding of the metabolic changes during transplantation, but also underscore the importance of further investigating the precise role of adenine and its implications for monitoring and managing ischemia events. In conclusion, our study sheds light on some of the metabolic changes that occur during DCD. We observed a decrease in fatty acids and a significant increase in sugar and amino acids, particularly alanine, over time during perfusion. These findings represent an exciting first step in understanding the underlying biology of DCD. Moreover, they have the potential to lead to the development of novel diagnostic tools that can improve decision-making in transplantation and ultimately increasing the availability of